In this lecture, we're going to talk about blood typing. In humans, blood type is determined by several different types of proteins that are genetically determined that are found on the surface of blood cells. So an antigen is a substance the body recognizes as foreign. These are things that can be attacked by the immune system. Antibodies recognize things that are foreign when they're in your body. So your body makes antibodies against antigens. Blood is typed by using antibodies that will cause blood with certain proteins to clump or do something called agglutination. So that's how we determine blood type. In humans, there's actually over 30 common red cell antigens, so those are proteins that are on the surfaces of blood cells, but the most vigorous transfusion reactions are caused by the ABO and the RH blood groups. And so those are the ones that are the most important if you need to get a transfusion. ABO blood groups give us 4 different blood types. There's type A, type B, and if you have neither of those proteins on your blood cells, you have type O, and it's also possible to have type AB blood, where you have A and B antigens on the surface of your blood cells. So the different types include AB blood, so that's when you have antigens from the A protein and B protein on the surface of your blood. If you have just the A antigen on your blood cells, you have type A blood. If you have just the antigen B, then you have type B blood. And if you lack the antigens for A and B, then you have type O blood. For transfusing, your blood type's important. If you have blood type AB, you're what's called the universal recipient because you can receive a transfusion from A, AB, B, and O. If you have type B blood, you can receive a transfusion from someone with B or O. And if you have type A blood, you can receive transfusions from A and O. And if you have type O, you can receive only O from blood, or only O from another O donor, but you are the universal donor. So O blood can be given to anybody. So we can summarize all of that in this table, right? So if you again have type AB blood, it means that the A and B antigens are present on your blood cells, and it means that you can receive blood from all of the other blood types. If you have type B blood, you have the B antigen on your blood cells. Your body will reject type A, but you can receive transfusions from B and O. If you have type A blood, you will respond negatively to B antigen, but you can receive blood from A and O. And if you have type O blood, you are going to react to both A and B negatively, but you can receive blood from O and donate to everyone. The other major factor that shows up on the surface of blood cells is this RH protein. We call it the RH factor sometimes. It's called RH because it was defined originally in rhesus monkeys. That's where the protein was first discovered. So the RH protein, you either have it or you don't. So there's not more than 2 types. Rh positive people have Rh proteins on the surface of their blood cells, and Rh negative people have no Rh proteins on the surface of their blood cells. And so if you donate Rh positive blood to someone who's Rh negative, they can have an immune response. You can have issues during pregnancy if a female is Rh negative and the father of her child is Rh positive. If the child inherits the Rh positive factor, the first child usually does not have a problem, but the mother, if she has a second pregnancy and her immune system is sensitized to Rh positive blood, she can have an issue with her pregnancy. This can easily be prevented, however, with something called the RhoGAM shot. It prevents the buildup of anti-Rh positive antibodies, and so then the pregnancy can carry on normally. The way that it works out, again, the first pregnancy is usually normal. The second pregnancy can be problematic because she's been sensitized to Rh positive blood, and so what can happen is that the newborn has hemolytic disease. But again, this can be prevented very easily by the RhoGAM shot. We do blood typing in order to determine what blood type somebody is. And so you mix all of the different blood types you want to test with an anti-A and an anti-B serum, and that allows you to determine what the blood type is. And this occurs because coagulation or no coagulation occurs. So when you do blood type, For Rh, it's the same thing. And this way you can quite easily determine blood type. Here's what a positive and a negative test looks like for each of the different blood types. So if somebody has type AB blood, it means that A antigen and B antigen are present on their blood cells. And so when you mix their blood with anti-A serum, it does this thing called agglutination where it gets kind of clumpy. And it does the same thing for B because they have both antigens. If you have someone with type B blood, the type B blood will not react to the anti-A serum because there is no A protein in their blood. And so the A result is going to look like this. It's just going to look like a mixture of a liquid. There's not going to be any clumps. But the B protein will respond to the anti-B antibody, and so then you get agglutination. So then someone with type A blood will agglutinate in the presence of anti-A antibody, and it will not do anything with anti-B. And if you have type O blood, you shouldn't see a reaction for either type A or type B antibody because there is no antigens on those blood cells to cause a reaction.